It's a pleasure to actually have the chance to talk to people from the University of Guelph. I know a lot of researchers from there, especially a lot of ecophysiologists. So I admire the research that they do there. So it's a real great pleasure to present this talk. And before I begin, I also would like to acknowledge the Indigenous people where this work was conducted as well, and where I'm currently presenting today. So I'd like to acknowledge the Darug people, the Aboriginal owners of the land where I am speaking to you from, and pay my respects to the elders past, present, and emerging. Now, the talk that I'm going to be presenting to you guys today is on Chytridiomycosis and mechanisms of skin disruption. So this is kind of a different perspective of looking at how chytrid fungus is disrupting amphibians all over the world. All right, so you probably heard a lot about the chytrid fungus before, but I'll give some background information about it and my approach to understanding this disease. So amphibians all over the world are declining, and around 43% of all known amphibian species to date are considered threatened. And around 2,500 species have been declining since then, and especially more rapidly in the 1980s, around 435 species have been declining quite readily. Now, there's a lot of potential explanation for these causes, and that can be kind of summarized into three different flavors. The first is habitat destruction, which is a very large component of a lot of amphibian declines. So if you see here on the world map, colored in green, a lot of the declines associated with habitat destruction can be seen around the coastal regions of America, in Europe, and Southeast Australia as well. But another important factor is overexploitation. So this is colored in blue. So this includes pet trade, medicinal purposes for food consumption. And this can be seen in Asia and Southern America. But a large proportion of these amphibian declines are actually classified as enigmatic. So these are not declines associated with habitat destruction or overexploitation. And you can see this is kind of colored all over the map. So including North America, South America, Central America in particular, parts of Asia, and also in Southeastern Australia as well. Large declines associated with these enigmatic causes. And because they're not usually associated with human disturbances, they tend to occur in areas outside of urban areas, human influences, and so on. So these can also include pristine montane and mountainous regions, such as the cloud forests in Central and South America, and also the rainforests of Queensland and New South Wales as well. There's a lot of potential causes for these, and researchers have been kind of examining these enigmatic declines and what are the causes behind it. And that can include the introduction of species that can either outcompete these native species, or predation, all sorts of different factors, or introduction of diseases as well. Pollutants, runoff, and pharmaceuticals have been all shown to influence or affect amphibians in some way or another that can contribute to their declines. Diseases, of course, which is the main topic of my talk. And of course, climate change. Global warming, increased UV radiation have all been shown to be associated with amphibian declines. Now, since the focus here is on diseases, I want to pay particular attention to fungal diseases. And this is because fungal diseases have been on the rise in the last century. So this is associated with plant-associated fungus and animal-associated fungus infections as well. And you can see here on this graph, there's a lot of number of these interacting factors increasing over the last few years, and in particular, a number of deaths associated with fungal diseases have been rising dramatically. And this also includes not only Chytridiomycosis, but also white nose syndrome as well, affecting North American bats, and a snake fungal disease, as a few example cases of these fungal diseases affecting our wildlife. But here I want to focus on Chytridiomycosis. So it's a fungal pathogen disease caused by the pathogen Chytridium dendropatidis. And we'll call it BD for short. but there is another sister group called Chytridium salamander variant, B cell, which has been associated with declines as well, that causes chytridiomycosis. But the current disease-causing pathogen is BD. Now, this example image that I'm showing you right now is a classic chytrid image that you would probably see in a lot of talks, and I'm including it here as well. This is an image that was taken by Joe Sartori in the California's Nevada mountains of amphibians, dead amphibians found in the early spring. So you can see some snow in the background here. This is a very classic apocalyptic type of image you'll see with these news outlets. So chytridiomycosis is, the BD, the pathogen that causes chytridiomycosis, is found all over the world. So you can see here in this world map, BD is basically found wherever there are amphibians. And it's been shown to affect over 500 different species of amphibians worldwide, and has caused some species deaths, so complete species wipeout for some of them as well. And it's considered the largest known decrease in vertebrate biodiversity caused by one single disease, right? So this is a very devastating fungal pathogen. So a bit of life cycle about this fungus and how my study kind of evolves around this life cycle. BD's life cycle is pretty simple to understand, actually. There's actually two main life cycle stages. One is a motile zoospore stage, as you can see on top here. So if I can, okay, I can't, oh, here we go, I can annotate. So there's a free swimming motile zoospore stage that finds an amphibian in the water and it attaches itself to the skin. After that, it starts maturing, develops into a sporangium, and then eventually it will, so it basically sticks underneath the skin layer and develops into a zoosporangium, and eventually matures and discharges more zoospores out into the environment, and the life cycle continues from there on. And it mainly affects amphibians, not all over the skin, but in areas where it's constantly moist and wet as well. And so it's usually found on the ventral surface of amphibians and the foot, toes, and legs as well. And in tadpoles, usually the mouth part, because this is the only area that's keratinized. What they do is feed on the keratinized part of the skin. And so they attach themselves to the tadpole's mouth, but nowhere else. And you can see on the right side here, we have a healthy skin, nice and smooth, right? And the epidermis consists of this layer here, as you can see. When amphibians are infected, especially when they're highly infected, the skin becomes quite irregular shaped and either becomes thinner or thicker in some parts and becomes a total mess, as you can see on here. And you can see some arrows showing where the zoosporangium is typically held, especially in the upper layer of the epidermis here, right? Okay, so I'll stop my annotation and move on. All right, so what are some of the symptoms associated with chytridiomycosis? Well, on the left, we have a healthy frog, and on the right, we have an infected frog here that I took a photo of. They're roughly around the same size, but you can clearly see that the infected frog is a lot more skinnier and it has redder skin as well. And so there's a whole bunch of commonalities that you can see on here. You can see behavioral factors that are being affected, so increase in lethargy, decrease in appetite, and reduce in skin shedding as well. So instead of shedding their whole skin in one piece, they shed it off in little fragments, as you can see on this image here. So an infected frogs will tend to shed off their skin in small fragments, as you can see on the bottom left GIF here. Morphological damage, they have increases in ulcers, hemorrhaging, I mentioned before, reddening of the skin, especially in the ventral area, and peeling of the skin as well. Physiological responses, they tend to have reduced skin function, altered immune functions and low blood sodium and chloride levels. And this is particularly important to emphasize for this talk, because this is probably the reason why they are dying off, because of the low electrolyte levels in their body that some researchers have proposed leads to heart failure, and that's why they expire from this disease. So the focus of my talk will be on osmoregulation in this aspect. But because of all these commonalities in responses across different species, I actually wanted to know, well, how common is this? So I actually did a meta-analysis on combining all these different biological disruptions from BD infection and kind of summarized it in one graph. So what I did was collect 304 effect sizes from 68 studies that looked at biological disruption in one form or another. And this consists of 52 species. And this is kind of a summary of the disruptions that BD might cause. So what this image is showing, or what this graph is showing, is that we have effect sizes. So this is the effect of BD on a certain biological trait, whether it's thermal regulation, skin integrity reduction, osmoregulation, and so on. And so a negative direction typically represents a detrimental effect, right? But for certain traits, such as hormone levels or energy metabolism, an increase or decrease in response to BD infection doesn't necessarily mean it's detrimental or positive. So in this respect, these kind of traits are represented as just either an increase or decrease in response. Okay. So what we have here, we can summarize all these responses in this way. Energy metabolism, so this is metabolic rate, oxygen consumption, CT max, and so on, tends to generally increase with BD infections. So as animals are being infected by BD, their energy metabolism increases. While on the other hand, on average, body condition, osmoregulation function, and behavior tends to decrease with BD infection. Okay. All the other traits tend to have a mix of results. They tend to spread widely, either an increase or decrease, and that's why you don't see a big difference here. So that's the extent of the biological damage, the current state of knowledge of biological damage of BD infection. But what I'm curious about is the mechanisms behind this disruption here. And so because BD only attaches itself to the skin and mainly affects the skin function, we want to understand how this decrease in skin function is affecting amphibians as a whole. Okay, so amphibian skin structure and function is a multifunctional organ, right? So they use their skin for respiration, exchanges of oxygen and CO2. They also have a mutual benefit with symbiotic microbiomes as well. So there's beneficial microbiomes on their skin, including bacteria and fungi. They especially use their skin for water and ion exchange as well, particularly sodium and chloride ions. The skin also acts as an immune defense as well. So they can secrete antimicrobial peptides to counteract any potential pathogens that's infecting the skin. It can be a physical barrier as well. So it can be used to protect against UV radiation, abrasion, mechanical stress, desiccation, and so on. And it's also used for acid-base regulation as well. So exchanges of hydrogen and carbonates as well, but to a lesser known extent. So the skin itself can truly be called a multifunctional organ. And so any disruption to the skin will cause a disruption to all of these different functions and protective features as well. So it's critically important for amphibians, and why this disease is so devastating as well. Now, amphibians are considered bimodal animals because they live in both freshwater and terrestrial environments. And in freshwater, their osmoregulation can be quite challenging because in freshwater conditions, they are more likely to lose these ions, these electrolytes, into the body, out of the body through diffusion alone and influx of water. So they gain water passively just by being in water itself. And on land, they're more prone to desiccation. So water loss, dehydration is a major factor of osmoregulating on land. And some frogs are able to deal with this quite okay because they can secrete these protective layer. These are lipids. So these are, that can also act as a sunscreen as well for frogs, especially tree frogs. And so they wipe themselves with the secreted layer to protect themselves from evaporative water loss. Okay. Now, in water, I mentioned before that amphibians will lose lose ions in the water due to diffusion, right? So in this example case, animals from the inside, so high concentration of ions on the inside will diffuse outside passively. And this is an example case of sodium ions, right? And this is because this is allowing the ions to equilibrate in their environment. But amphibians are able to maintain their ion levels inside their body by using some sort of active transport on their skin. So they can actively take in ions from the environment into their body through these active transport systems. And why I want to discuss this in detail is because this is where we can understand this is one of the factors that I think is important for understanding how chytrid fungus disrupts the skin function. This might be quite a lot to take in for a lot of non-ecophysiologists. So I'm keeping this very straightforward and simple by just getting you guys to just paying attention to sodium transport. What I want to emphasize here first is that the skin function can contribute to all sorts of different ion regulators. So potassium, chlorides, hydrogen as well. It's all being exchanged through the skin. But here I want to focus on sodium transport in particular, because this is the main chunk of electrolytes being exchanged through the skin. So on the outside, so the top part represents the freshwater, the outside system, and the inside represents the animal itself. So what the amphibian needs to do to exchange sodium into the body is, first, they can exchange sodium passively into their cell, in the cell membrane, because there's low levels of sodium inside their cells, and they can exchange that with potassium, which is very high within the cells. And so this is through the epithelial sodium channel, which is called an ENAC. And on the basolateral side of the cell is the sodium potassium pump. And this is the important part. What they do is exchange energy to move potassium and sodium across their against their concentration gradient, and this is why it allows sodium to be transported into the body through this fashion. Okay, so these two transporters are the main targets for what I'm looking at for this research. Okay. Now I want to add another important component to this, because I mentioned before the skin is a multifunctional organ, so keeping the skin healthy and functional is a very important factor in this. And originally I wanted to play a video within this PowerPoint, but apparently this media cannot play too easily. So what I'm going to do is share a different screen with you now. Okay, so hopefully you can see this video here, and I'm going to show you a video of a frog shedding its skin. So the frog will shed its skin because it's trying to keep the skin healthy and functional, right? You're removing the old skin so you can rejuvenate the new skin underneath. And in this image, in this frog video here, we have lipstick that's being put on the frog to visually indicate how the skin is removed. This is non-toxic, so it's okay for the frog. You can see that this old skin is being removed. It's being split in half along the body. And the frog is using various postures and limbs to kind of move the slough skin into the mouth to be eaten. And this is shown to be a beneficial process for the frogs, not only in nutrient uptake as well, but it removes potentially harmful pathogens on the skin. So this behavior is the first line of defense for amphibians. I'll stop sharing the screen now and go back to the main presentation. Okay. Why I want to emphasize this is because my previous research has shown that the act of sloughing itself also causes disruption to the osmoregulation temporarily. So that act of removing the dead skin and rejuvenating the new skin causes some disruption to their ion exchanges because their skin is not fully developed then. And so the act of skin shedding itself may actually be a double-edged sword for animals that are infected, because Chytridiomycosis is affecting osmoregulation, right? But so is sloughing at a certain time point. So the act of trying to remove the fungus may actually be exacerbating these disruptions to skin function itself. And so I wanted to find out what the extent of this is and whether or not this is the case. Was my original hypothesis for the study, and I wanted to see how it disrupted the skin function. So the aim of the study was to first determine the mechanism of cutaneous ion disruption on the skin and test whether sloughing or not exacerbates this ion disruption when infected with BD. I also wanted to look at the energetic cost of sloughing as well, so how much energy they expended during this behavior and whether the chytrid infection contributes to that or not. I may present that depending on how much time we have. And I approach this from an integrative perspective. So using whole animal responses all the way down to gene expression responses. And if we do have some time, I may also present some species level responses as well with a collaborator. But this work was done during my PhD with my supervisors, Craig Franklin and Rebecca Cramp from the University of Queensland. And for the integrative response, I used one species, Latoria cerulean, as my model species. As you can see on here, it's a very beautiful animal. This photo does not do it justice, but it's a beautiful animal to work with. They are considered, they are still common in Australia, but they are susceptible to the chytrid fungus. So we're not working with an endangered species here due to the kind of approaches we're using. So the first one is the whole animal response. And the question was, how much ions are being lost through the skin when infected with BD? We actually don't know how much ions was being lost originally. Previous studies have looked at other approaches, but using whole animal responses, we can get a better idea of the extent of ions being lost. So what we do is measure the conductivity of the water that they're in. So we place them in distilled water, right? So there's no ions in the water. So you would get a conductivity quite low. So this is the baseline levels on here. And when you put the frog in, you can see a slow increase in conductivity. So that means they're losing a little bit of ions while they're in the water, but not so much. And this is what happens when their skin's sloughing, right? There's a huge increase in ions being lost when they shed off their skin. And after they shed it off, it remains quite stable afterwards. So we are able to place video cameras and watch them while they're sloughing. And so we can get accurate times of when this behavior is occurring and when this dramatic increase is associated with that. So when we combine all this data together, when they're infected, we can get this graph here. So this is a data showing when they're non-sloughing. So this is the first image here where you just get baseline levels, right? So on the x-axis is infection intensity. So this is how infected an amphibian is. And this is log mass-specific conductivity readings. So this is controlled for by body mass because larger animals may just by default lose ions more readily. So it's controlling for body mass. And you can see on here as infection intensity increases, their rate of ion loss also increases as well. And so this is a really significant finding here, but not particularly novel in this case, because we're kind of expecting this. But when they're skin shedding, we actually don't see any effect of BD infection. When they're infected with BD, you see a very similar response here. And this is because they're already losing a lot of ions when they're skin shedding, right? But when they're infected with chytrid fungus, they seem to lose the same amount when they're skin shedding. So this is quite interesting. I wasn't expecting this. I would expect it to continuously increase as well, as you see with the non-sloughing. We can get some hints of why this might be the case by looking a little bit deeper into the skin. So what we did was take samples of the ventral skin. This requires euthanasia, of course, but we're able to measure how permeable the skin is and how much sodium is being transported into the skin, right? And we can do this by putting them into this chamber called an Ussing chamber, which you put a piece of skin on two sides of a chamber, and you have one side that represents, you put solutions that are the same as the frog's internal solution. So we call that a ringer solution, and the outside is just fresh water. We put the other side as fresh water, and we can see how much sodium is being transported across the skin by putting a drug called amiloride. Amiloride blocks the sodium channels, so prevents any sodium from being transported into the skin. So we can see the relative contribution, we can see the change in the skin function based on that and deduct how much sodium is being transported based on that. So what this graph is showing is that intermolt is when they're non-sloughing, right? You can see that when they're uninfected, their sodium transport is around this value, and when they're infected, there's a decrease, right? So there's not much sodium being transported back into the skin when they're infected. What we didn't anticipate was that they actually increase in their sodium uptake when they are sloughing. And that could explain why you don't see a difference in the amount of ions being lost when they are sloughing. It might be because they are actually trying to actively uptake the sodium channels, the sodiums back into the body for that. And I'll give you some explanation for why this is the case when we dive down later on. But to further exemplify this, that the disruption is mainly caused by the skin function being disrupted. This is the skin resistance here, which is showing how much resistance there is to leakage, essentially. So the lower the resistance, the higher the leakage. And when animals are infected, they have a high leakage. And this is especially the case as well when animals are sloughing, they have increase in leakage. Makes sense. I've seen this in my previous study with cane toads as well. But when infected, it further decreases as well. So they're losing a lot of ions during sloughing, but they're trying to uptake that back on with these sodium transport systems. Okay, so we see a decrease in sodium uptake when they're non-infected, and an increase in sodium uptake when they're infected. So what's happening down at those protein levels, those sodium-potassium pumps and the epithelial sodium channels? So first focus on epithelial sodium channels. And this is the abundance of sodium channels in their skin. So this is measured through Western blotting methods. And this is the expression. These are just gene expression methods of measuring how much ENAC is being expressed. So we can see here that ENAC is decreased when it's infected. So there's lower numbers of ENAC on the skin, but there's an increase in expression. So what this is suggesting is that the frogs are actively trying to make new ENAC proteins, but there's some sort of translational disruption happening from that increase in expression towards the increase, towards the production of those proteins. When they are skin shedding, we can see that, okay, it's a similar trend as when they're not sloughing, just to a lesser extent here. So nothing too important to discuss about in this aspect. But when we look at NKA abundance, so this is the epithelial sodium channels, we see there's no differences in the number of sodium channels on the skin when they're non-sloughing. But when they are skin shedding, there's a huge increase in abundance when they are infected. All right, so when they're skin shedding, there's a huge increase in abundance that's associated with that increase in uptake back into the body, and also the no difference in sodium ion losses through the skin. And the expression is seen similarly as well with the ENAC. There's also an increase in production of those sodium, the NKA gene expressions as well. So that's a really interesting finding here. We also looked at tight junctions as well. We didn't not only looked at the exchanges of sodium ion levels, but also looked at the junctions itself. And this was done in collaboration with Julia Goldberg, who was from the York University in Toronto. And she was a master's student who did an exchange here with us. And she looked at the aspects of tight junctions. So what tight junctions are these little proteins that keep the skin intact, essentially, prevent leakages. And what we saw with Chytrid before was that there was an increase in leakage with the skin. And so what she was interested is which one of these proteins were disrupted, right? And she used a similar kind of design to an Using chamber, but used this experimental chamber itself. I forgot what the name was called, but essentially you have the skin placed on the side here with a solution on the outside, which was fresh water, and a solution on the inside, which was, what was it? Ringer solution as well. And what she did in here was she put dextran, which is a sugary substance in here that usually doesn't permeate through the skin unless it's disrupted one way or another. And so you can actually extract liquid from the top chamber. If there is any leakage, you would see some of these sugary substances being leaked out of the skin. And what you can see on here is that as animals are infected, you can see here when animals are not infected, there's no there's none of that sugary substance being leaked out, which is good. But when they're infected, you can see this huge increase in leakage. All right, so this corroborates with the previous findings that we had. And she also looked at the same kind of procedure I did as well. So she did some histology work, some Western blotting work, and some gene expression work. And I'm just going to present to you one of the examples here, which is oculin. And you can see oculin is kind of distributed on the basolateral side of the epidermis along here, and with the abundance as well, same with ENAC, and you can see a decrease in oculin abundance. So the skin is not as tightly formed because of that decrease in oculin, and also an increase in expression as well. So this is a great finding because this was done independently to me. I had no contribution to this other than giving them the frogs, but she did all the study herself, and she found very similar trends to what I did as well. So it's pretty confident. It's good to see this kind of similar results between us as well. So in conclusion from this integrative aspect is that infected frogs tend to have higher cutaneous ion loss from what we saw with the whole animal responses down to the protein abundance as well. And sloughing does not further exacerbates this ion loss, which is a good sign. So what that means is that sloughing is probably okay for the animals. It doesn't further exacerbates the ion loss that is seen. And BD is shown to reduce or inhibit regulatory transport proteins, so these ENAC and oculin as well, but doesn't seem to be doing so with NKA, and it affects tight junctions as well. So what this study kind of concludes is that it provides a mechanistic explanation for disruptions in skin integrity itself and the regulation of this disruption. What this also shows is that sloughing may provide some temporary restoration to the skin function, as you see with a huge increase in ENAC abundance. But the problem is that this function is only temporary, because the BD can actually reinfect the skin and further decrease the NKA abundance back down again. So it may actually accelerate this disease progression for susceptible species. For species that can remove the fungus quick enough, there won't be a problem. But for amphibians that have this kind of disruption, continues to be disrupted, this may actually hinder, sloughing may actually hinder their ability to regulate the skin. And since we do have some time left, I'm going to continue on now to present some of the studies conducted with Michelle Omer on a broader perspective now. So now we're not looking at the physiology, we're looking at more of the behavioral aspect. And this is done with interspecific comparisons. So we've got a whole bunch of Australian frogs here, and what I'm trying to emphasize now is that the act of skin sloughing actually decreases BD load. And you can see here with a lot of the amphibians that are infected. So this is before and after. Red is before and blue is after. You can see for all the amphibians, there's generally a decrease in BD load after the animals are infected. We can kind of see this in another approach as well. So the relative percentage difference you can see here. So almost all the amphibians here a decrease in BD infection, BD load after they slough. With some species like Limnodynastes tasmaniensis, sometimes they can remove up to 100% of BD load. So they can remove it completely and they're fine. But there's a weird case of clinical versus non-clinical. So these are ones that develop chytridiomycosis, ones that don't. So depending on how much BD is removed, they may either continue to increase in BD load, while others see a different response. They can maintain a relatively low load after they've been exposed. And the reason why this is the case is because BD itself isn't always on the skin surface. It's also found slightly below the skin surface as well. So we've got an uninfected frog up here. You can see here, it's nice and clean, as we've seen before. A frog that has low infection load generally has and does okay with the skin as well. So I'm just going to annotate. It's quite nice. The skin is really nice and smooth. When they have very high infection load, you can see the zoospores here and the skin being sloughed off in small pieces, right? So some of the time they can be sloughed off with the skin, and that's why you see a decrease in BD load. But you can sometimes see that they're still hiding underneath the skin when the skin is sloughed off. So they're not always removed completely in this case example. And I also mentioned as well, sloughing is an immune function, immune response as well. And what both Michelle and I found separately is that frogs increase in the rate of skin sloughing in response to BD. So they actually increase their rate of skin shedding in response to BD infection. So you can see here in Michelle's graph that when they're infected, they tend to decrease the intermode interval. So they shorten their interval, means they slough more for a period of time. And also the same with mine when they're infected. The more they're infected, they slough more often. And because of that disruption in skin function during that period, that increase in skin sloughing rate further exacerbates that increase in iron loss for that reason. So this is contributing to their disruption when they're at very high infection load. The increase in sloughing rate. I mentioned before, if we had time, we could go over some metabolic costs as well. So measuring oxygen consumption, or sorry, CO2 production in this case example. And so we do have time, which is nice. So what I'm presenting to you now is what we've seen similar to the iron loss, but here is CO2 production. So on the x-axis, we have infection intensity, and on the y-axis is mass-specific rate of CO2 production. So as the infection intensity increases, you see an increase in metabolic rate or increase in CO2 production. And same thing with sloughing, but not as high to an extent. So they still increase in CO2 production, but not to the same extent as when the non-sloughing as well. So similar findings to what we had with the iron loss. There's also a body size effect as well. So smaller frogs tend to be more susceptible. You see die-offs in the wild associated more with smaller individuals. And one potential reason for that is, well, of course, the increase in iron loss per surface area as well, but also with the energetic expenditure. So CO2 production tends to be a lot higher for smaller infected animals. So they have a slope that is much lower, meaning that the slope is a lot greater for small animals. Note there is a sample size of 1 for small animals. It is very hard to get CO2 production from a very small animal with the setup that we had because it increases evaporative water loss very quickly. So it's very difficult to get these behaviors at these specific times, but so be wary of these kind of results at the lower end. With sloughing, we can see it does increase in CO2 production, but when they're infected, there's no difference here. And we don't have any infected frogs. Very small infected frogs are sloughing because, again, it's a very hard behavior to quantify in this case. So what we can conclude from these additional experiments that I presented is that sloughing is an overlooked but a critical important aspect of cutaneous functions in amphibians. A lot of these studies with chytridiomycosis don't consider sloughing, and sloughing itself, as you can see, can interact with how you perceive these disruptions with amphibians that are disrupted by chytrid. Sloughing does help with increasing, clearing some of the infections as well in some species, which is good to see. But BD impacts both cutaneous regulation and in skin integrity, as we've seen previously. And the increase in skin sloughing is definitely energetically costly on top of infection disruption. So if they're increasing in the skin shedding rate, they're also increasing the amount of energy to do that behavior as well, since you can see an increase in CO2 production. What are some conservation implications from this, though? You know, I've talked about these mechanisms of disruption. How can these kind of results contribute to better management of these frogs? So it's all involves around sodium transport, right? The loss of electrolytes. Some studies have tried to introduce salt treatments that have been shown to improve host survival. So what they did was have these giant mesocosms outside in a wetland somewhere, and they start putting in some salt into the environment, infect the animals with chytrid, and see how these frogs survive. And they found that with salt treatments, the frogs that are in these kind of treatments tend to survive better than frogs that are not in these salt treatments. There's some caveats with these introductions, though. You don't want to deliberately introduce salt into wetlands because that can have unintentional consequences on the wildlife around them. You know, a lot of animals and plants rely on fresh water, and introducing salt may disrupt their osmoregulations indirectly, especially for frogs as well. So this is probably not the most ideal treatments, but has been shown to increase survival. If animals are critically infected, so they have very high BD load and almost at the brink of death, Jamie Voiles in 2009 has shown that introducing electrolyte treatments can prolong their life recovery, but they still die off eventually at very high infection loads. But at low infection loads, supplements of electrolyte treatments actually help them recover from chytrid fungus. So we could possibly establish a critical period, a possible threshold of BD infection that may allow them to survive and recover before the onset of the disease, because the disease only occurs when they are at very high infection loads. Okay. We also need to be carefully considering of smaller young individuals. We need to pay focus on that because they tend to be the most susceptible to iron losses and increases in energy expenditure as well. So need to pay particular attention to that. Great, we still have some time. So this is the last slide that I'll present. I mentioned before, we could possibly establish a critical period for determining whether or not it is viable to save animals from BD or not. This is an example case here with zoospore load. So increase in zoospore load and sloughing behavior, right? So you can use this as an indicator. So normal sloughing behavior, the act of shedding off the skin, generally can be seen here up to a certain infection load. And then after a certain period, they become so lethargic that they just stop sloughing altogether. So the increase in skin shedding rate, right, when they're infected, but up to a certain point, they will just stop altogether. It's just too costly for them to do it, or they're just too lazy, lethargic to do it, and they just stop altogether. And you can use these bimodal models to establish, to predict at what threshold would you expect to see them to stop sloughing. So a 50% threshold for 20,000 zoospore loads, you may expect to see 50% of these frogs to stop shedding altogether. Instead, they'll shed off into little fragments. So you can use this kind of threshold to establish, okay, what BD load is important to consider when trying to help recover these amphibians. After that, it's probably no point in helping them recover because they're at a point where they can't be recoverable. So that is it for my talk. I'm happy to answer any questions for anyone. There's a lot to take in, wasn't it? There's a lot of results to present there. Hopefully I didn't go through them too quickly, but there's plenty of time to answer questions if there's stuff that you want to clarify on.
I wanted to just jump in, and before, because I certainly have some questions, I imagine some others do. And Nicholas, I wanted just to say, like, give people an opportunity to hammer on their applause emoji or do it in real life if they're comfortable turning the cameras on, because that was splendid. Like, just kudos to you, kind of a fantastic demonstration of an integrative approach, like taking from physiological ecology, from tissue to species to conservation, and even leaving us with some management things to consider. Like, so, and doing it with some of the most beautiful slides I think I've ever seen. So,
thank you.
Done. Well done.
That's good to hear.
The chat is open if people are somewhere where they want to throw in, and I see Catherine has her hand up. Let's start there. And I will do the thing.